Today I would like to again present some ideas or my vision on challenges which we have in space bioscience. We're working already, I believe, hard to address some problems which were identified for us for manned space exploration. This primarily with primary goal to identify fundamental problems in response of living organism to space flight environments. Second, we would like to accumulate knowledge about underlying mechanism with such response, to apply it to develop a countermeasure which to support presence of human space for a very long time. And this is actually our primary goal. And third, absolutely obvious is that if such kind of knowledge will be accumulated, we can apply it to improve life on Earth in many areas like pharmacology, oncology. It would be different medicine and also technology for development of health countermeasures. In spite of many challenges exist, I would like probably first to talk a little bit about our position, how we consider space exploration, because over, like I mentioned many times already over last year, space exploration program was under some kind of re-evaluation, because public requests, they want to know where we are in terms of our plans, how reasonable our plans, how they ambition and how they fit to the vision, long-term vision of society in terms of space exploration. And this summer, NASA was very democratic, asking National Academy of Science to provide their opinion about where we are in terms of Mars mission. And the response was a kind of surprise because National Institute of Medicine released a report in, I guess, in April 2014, which said that probably we're not ready still to go to Mars, saying that our health standards or NASA health standards not yet fit to ethical norms of society. So people can interpret this as, well, we have a problem to provide safety for health of astronauts on their mission to Mars and return back. And this is very big challenges, actually. Public and communities are thinking that what we were doing all of this time, whether we're going in the right direction, how we can provide or ensure safety of astronauts. And last summer curiosity data, like I already mentioned, about regarding radiation on our way to Mars and back, show that radiation level would be higher than we expected. This means that potentially astronauts can come back with some health problems. And society, in this case, NASA in particular, would be responsible for that they did not provide all of this. This is why just community have rights to challenge NASA on this issue. They want to be ensured. And also we again coming back to how enthusiastic of our society regarding space exploration. Same as it was like 20 years ago, or it's like you ask me question now, how enthusiastic we are, what would be 10 years later. And we should admit, and next slide I make some kind of reference, which saying that if we ask people regarding space exploration, we probably will get some kind of disappointing answer, which was expressed in many good thinker in this world, that yes, we probably lost some kind of interest in space exploration, and we need to know why, because NASA completely focused on actually to reach the goal. And if public somehow disappointed or don't believe, we have a problem which we should solve. And after this report of National Academy of Science was released, NASA Administrator Charles Bolden released some kind of statement which really was like that while Mars is our goal, we recognize this capability in space-faring nations today are not sufficient to safely land and return humans from surface of the red planet. As well, we have gaps in our scientific, engineering, and technological knowledge. This actually gives us some kind of idea that yes, we should kind of reconsider our gaps and put more efforts to provide safety for astronauts. And again, why we need it? Because more and more people which recognize leaders in science, politics, economy, start to admit that I'm concerned about survival of human race in general. And even such person, which we know, Cambridge physicist Stephen Hawking, perfectly put it in such a way which probably, I would say, not recognized in real everyday life. His statement was that, question like that: we're in a small planet, and this planet exposed to big danger in statistical cosmological events. And the human race is only nest for human race. And probability that someday we will be hurt asteroid or something happen else in cosmological kind of scale, it's very high. And we can just kind of vanish from the universe. At the same time, we understand that really, if we would like to protect human race, we somehow should look outside of Earth or establish some backup somewhere in terms of colony on a Mars or another planet, or somehow to preserve it for better time. I don't know in cosmological scale what is better time, but I have some idea how we can just kind of proceed in this direction. What is also necessary to bring, again, do we have solution for that or not? In previous years, space enthusiasm was so high that was kind of every day in newspapers. Now, of course, you can just rare to see something about space exploration. Of course, we know that continuous research on International Space Station, we can see every day. We have channels, we can go to a website and real time to see what they're doing there. Potentially you even have possibility to have a conversation with them. But it still not looks like for me what we had before, when people put so much effort and enthusiasm saying that we have so ambitious plans. And you remember, even just at NASA, I remember at NASA Ames Research Center in 1970s, they create so ambitious plans, and young people, students, putting some kind of proposal, like 240 pages proposed in details, which show how they want to proceed with space colonization for orbital flight, for example, and also Biosphere project in America. It was amazing actually at that time because it's addressed so important problems which now some of them, of course, already executed in the International Space Station because it's close environment which provide conditions for successful mission operation. Also idea about how we can use asteroid, for example, how we can make from asteroid spaceship, which would be actually for us as another station. All of this plan was so ambitious, yeah, that now we trying to review them, and they look like very fantastic for us in this kind of time. And it's a little bit kind of disappointing, but it should be opposite. If you read newspaper, see movie, everywhere we already have information how we can explore Mars, how we should go to another galaxy, and what actually plans. And this ambition, people, who are these people who just kind of writing this? Is it just kind of space enthusiasts, or are they professionals? And they making picture for us, which for, even for me, as a scientist at NASA, as a driving forces. You know, I'm trying to be on the front of this, yeah, and I feel that I'm a little bit behind. I need to do something more than I'm doing right now. And this brings us to reality, that yes, reality is not so bright in terms of exploration. We have many problems to solve, which in previous years we thought that we will overcome in one moment, like that. Now, just if we're just talking about whether we're still going to proceed with we plan, it's again to make us a little bit confusing. Without new technologies and breakthrough progress in some areas, we probably cannot do this. And such breakthrough, like if we start to consider, okay, we want to build a spaceship. On current status of technology, this spaceship will be so big, like presented in this slides, it will be comparable with highest building in New York. It will be so heavy and so big that you know, it is unlikely that first of all we can build it in the next 30, 40 years. And second, it would be unreasonable to do this. Maybe better to proceed with waiting strategy. Let's just sit, wait, and see how progress and technology will proceed. Probably at that point, 30 years later, we will be better positioned than build this ship. And it will be old already at the time when we're going to launch it. And that actually always a problem for NASA. Technology develops so fast that you know, we cannot incorporate it immediately. Come to the point that yes, to build some starship probably will be not reasonable now. If we start now, we will finish in 2000 ever, whatever. And it will be already old, maybe, because we will using current technology to build. For that time, it will be already something new. So we need to come to probably use some new ideas, and it's presented in this slide as a Alcubierre warp drive. It's a Mexican scientist who in 1990, I guess, for proposal that we can actually potentially use new technology or completely new vision, how we can warp space-time and so proceed with much or close to speed light to explore even closest constellation or Alpha Centauri in weeks. Of course, nobody answered. He's using interferometer to justify or just kind of proof, demonstrate proof of principle that it's possible. And all science and the physicists working on this is saying, yeah, it's possible. It's surprising, but it's possible. So if we would get such kind of technology, even just probably some operational model in next 10, 20 years is probably better than if we put efforts to such kind of research that build a very big, old already spaceship. Anyway, when we're coming to problem of manned space exploration, we also balance this space exploration with robotic exploration. Since Apollo and since we just sent human to orbit, it always balanced. And this balance actually probably related that to economy, to financial capability, for scientific engineering capability. Sometimes it's easy to spend a robot, and it's very successful, provide information so humans kind of never can get, and it's very fast, efficient. But there are some moments when presence of humans absolutely necessary, not only in decision making at that moment, because some moments very could be crucial, and the robotic mission would be not able to solve it. Also, you know that even for Mars mission we have a delay in like almost 40 minutes, yeah, in this kind of events. It's not real time. If something really, let's suppose something happened with astronauts, and there will be operation, right? Surgical operation, yeah? And we will follow instructions from Earth, very professional medical doctors, yeah? And you astronauts, which of course not perfectly be trained as a surgeon, yeah, will do some kind of surgical operation. Well, you know, if you open and make some cut and you see something only like 20 minutes, you need to get a response from Earth, and it will come, and maybe it's already too late. You know, it's actually, you need to kind of close everything. This problem actually also always should be considered, that we not in real time, even in such kind of mission which we believe now close. Mars is already in our mind very close, like Moon to us. We need only to get there. As soon as humans in space, physiological alteration so dramatic, and we know this from reports, from whatever we even see in astronauts, like puffy face, they becomes a little bit looking unusual, or sometimes we see how that the hair actually in low gravity, it's, yeah, very nice kind of picture. But something which we not used to see on the Earth environment, even just in small dive, which we're using to mimic low gravity. I would like to bring only two classical problems, which I believe that's very important. All these physiological alterations occur and could be handled. And they most important in some kind of transitional period. After adaptation over some time, most of them return to clinical level, and only two factors are always increasing. It's just bone resorption, muscle actually atrophy, and radiation. And this slide, for example, presents a linear increase. To what extent, we don't know, but question even is how reversible this changes. It might happen that, yes, they cross some line and they never can come back. There is no point of return. And these changes so dramatic, they remind what we have in osteoporosis here, aging osteoporosis, when presented in this slide, bone density drop so significantly, and we kind of a little bit, not a little bit concerned that some bone fraction may occur in astronauts. They even will not expect that it could happen in any moment, especially after probably trip to Mars and landing on the planet. This is one of the concern. And when they start to develop countermeasure how to prevent bone loss and muscle atrophy, best solution so far is exercise. But when you exercise, astronauts exercise, it's increase the metabolism, you know. And what is interesting, turns out, if they used food enriched with protein, it turns out the bone loss start to accelerate, becomes even more. And this problem actually now in solution, they handled it because it's kind of reasonably to expect. But initially when it came, people was kind of surprising that probably we not have solution, because we provide more energy for astronauts to enrich the food with protein, and suddenly this come in spite of exercise to accelerated bone loss. When we're going to Mars, of course, we can expect that bone resorption, bone loss will be so significant during flight to Mars. It's approximately up to 2% per month, depending on a bone. If you count this for 2 years mission, you can see, as presented in this slide, strength bone will drop almost 15% on our way to Mars. Then on a planet mission, it will be slowed down because low gravity of Mars, but then it's going again on our back trip to drop, and so finally we may have like 40% drop in bone and muscles. Yeah, this is absolutely unacceptable. I mean, probably astronauts can not walk on under condition of Earth's gravity. They need some kind of recovery, but recovery required much more time. For example, if you want half a year or 1 year in space, bone recovery require like 2 or 3 years. And this, if you in Mars mission 2-3 years, you can imagine how much time it's required to adapt again to Earth's conditions. Second problem, it's radiation again. So radiation risk we discussed many times, it still remains because we don't have technology which completely protect astronauts against radiation. Of course, we have some kind of shielding on the International Space Station, but it's not so efficient. We have a lot of very good technical solutions, but they're not complemented yet in NASA, and we don't have clear vision what of this technological solution will be implemented for Mars mission. Another problem, which I believe that is if we're going to stay in space, can we stay for a long time? For a long time, I mean, can we just spend their generation? Like, remember Star Trek generation ship? It's many generations occur over there. The question is that how it's happened. How in conditions of space flight, which environment, well, from this point, what we have now, so dangerous for human, how we can proceed to next generation? So question is it reproduction in space possible or not? So far, we have no evidence that human could be born in space and develop well. If question develop to what? Actually, certainly some kind of they adapt and the conditions being in space. So far we have Drosophila, for example, which is born in space. They return and they confuse how to fly. They need so much time on the Earth when they return because they were born in the space, how to fly. Some of them kind of can fly, another not. We have zebrafish, which also was kind of born in space. When they return back, they behave completely different from the group which was born on the Earth. They kind of lost orientation. They need a lot of the swimming in completely different directions. Something wrong. So they certainly, for them, it's not natural environment. And questions still remain. Can we just kind of at least to think about whether humans will be born in space? And probably not unless we solve problem with with low gravity. Artificial gravity also have a lot of problems. In spite of, let's suppose 100% we can solve and provide artificial gravity, but what kind? Usually so far we have, say, artificially centrifugation, right? You provide some forces which gives you impression of gravity. But in reality, we have a lot of problems because another forces presence over there which influence human vestibular and making so much confusion that you need to reorganize or have long adaptation for your brain to operate under such kind of environment. So artificial gravity, it's not like tomorrow or kind of next probably 10 years. It requires a lot of investigation and input. In my area, research, for example, also for medication, drug medication, to support life and good health astronauts, we need actually to have normal therapeutic treatment in the case of whatever, headache or simple or even more complicated health conditions. And we know from what we have over decades that some drugs or medication degrade in space much faster than in Earth. Like for 2 months they can lose 20-30% of potency. And we know that FDA requires, as soon as it lost 10%, well, they immediately remove from the market, right? How we handle this? Yeah, if for 3 months they lost already 40%, it doesn't make sense to take aspirin or Advil at all, because they probably will not affect some. And this is problem again remains. And so far, we believe that I still believe we can solve this problem. It's not big one, but still exists. Also, given to what I just said about physiological alteration, we're not even sure that some of medication will work in space in the same way how they work on Earth, because physiology completely different. Yeah, electrolytes, for example, in blood are different, and that may change pharmacokinetic and pharmacodynamics. So that's still remain a problem. At NASA, Human Research Program, it's one of the program which handle all this interesting question. Of course, priority is that Human Research Program should estimate risk for health of astronauts. This is priority, and develop kind of countermeasures which would provide some kind of safe environment for operation in space. Now I would like to change this subject and come to my favorite subject. It's about metabolism, human. I believe that I would like to advertise that probably solution which involve metabolic control may help to solve many medical and health problems in astronauts. I believe that it's human space exploration not possible even if we cannot have control on metabolism. Why? Because I believe, and it was demonstrated, that metabolism is important. I would say even just it's handle metabolism. It's important strategy for all life. If you consider, for example, evolution, yeah, during evolution, environmental conditions change so dramatically that if we would kind of be conservative, life would not exist on the Earth. And due to mutations, which is driving mechanism in adaptation to environment, yeah, to select whatever fit appropriately to current one, we can just have such kind of changes. But what if conditions change so dramatically that we cannot change DNA? I mean, cannot change genes, nothing. I mean, mutations not help us. So it's dead end. But nature developed another strategy. You can reduce your metabolism so dramatically that you sustain in some state, and the environment will not affect you. This slide, for example, presents hypothesis which was discussed in literature, and we really doing some experiment to support this hypothesis that if an environment changes so dramatically and physiology cannot respond to save life, dormancy state might help. And we have a lot of evidence in nature which demonstrate that yes, if you reduce metabolism to some dramatic level, under which response to environment minimum, you can preserve life. And I put here just some quotes from astronaut Pete Conrad, which kind of a little bit impressed me. Quotes like that: "I always thought that the most significant thing that we ever found over moon mission was that little bacteria who came back and lived, and nobody even said about this." He just referred to bacteria which was in a camera which they left on a moon surface, like approximately 1 year. And later they came and took this camera, and turns out that bacteria which was there survived all this year, complete vacuum, radiation, whatever, for natural kind of space environment. And that was absolutely amazing. And we found a lot of evidence over the last time that some life, if metabolism could be suspended, can be preserved over a long period of time. And here I present some finding in South Mexican in 2000 year. They found that bacteria which was a suspended kind of animation 250 million years can be returned and be alive. This is amazing. And also another, for example, 30 million years sleep of germ, they also can return in life. But if taking into account that 90% even more of Earth's mass is bacterial mass, and if from all this 90%, 98% are not culturable, they in some state which they're not alive, but they're not dead. It's kind of suspended animation, but potentially they could be cultured. We don't know even conditions just how to return, but with potential to return. It's not what we kind of heard about cryonics, for example, when they putting people in liquid nitrogen, right? And with hope that if it turns out and technology develops sufficiently, they can return it back. Well, no, it's kind of absolutely sustainable, normal, evolutionary kind of program when some life could be preserved and stay until some better conditions for their life will come back. And this related completely to what we have now. Recently, over last year, scientists demonstrate that many life creature can survive even deep space environment. Water bear, for example, small organism, microscopic, but still very complicated. It's not bacteria. Can survive in vacuum, liquid nitrogen, temperature like 360 degrees. It's kind of amazing, and return to normal conditions. This tells us that, well, maybe life can exist in space. And I believe that it's 2 months ago, it was one of the projects and for International Space Station when they start to taking probe from outside of space station to see whether they have biological contamination over there. Question is what kind of contamination we can have in space, like minus temperature plus huge vacuum exposure to radiation. But nevertheless, probes were taken. I don't know yet results. And also even more, scientists believe that bacteria or microbial life might exist in orbital space. Maybe in some kind of encapsulated in ice at low temperature, because temperature even in my flight was minus 52, right, and outside of plane. metabolism certainly will be decreased to some level that radiation will be not so dangerous for them. And so we're waiting results for this probe from outside to see whether any life may exist in vacuum. And that's very exciting. This is normal strategy to reduce metabolism for animal kingdoms. Yeah, these slides present variety of animals. They're very cute, which hibernate. And hibernation is very normal. If something wrong or conditions not so good, better to slow down and just wait to better conditions. Some animals, for example, here I present bear. Bears hibernate up to 7 months, and even giving birth to actually young guys in state of hibernation. And most important, they can delay or postpone a little bit these births depending on the conditions. Most important and amazing that during such kind of inability or sustained animation, which they never have experienced bone loss. You know, they not moving much, and they never experience muscle atrophy. Of course, they lost weight, but strong. The bones strength is very good, and muscle is good. And this means that there is some underlying mechanism which potentially, if explored, could be applied to prevent muscle and bone loss for astronauts. All these animals have some mechanism, and it's absolutely clear. One creature like dormice can hibernate 373 days. This is more than a year. Actually, you can say, and this is cute creature, you can see it's kind of small size. Nevertheless, 373 days, it's kind of, what kind of life is that? But nevertheless, they can very long exist in this state. And this is actually one of the topic and one of the main projects of my laboratory to investigate hypermetabolic stasis. And of course, we believe if we can incorporate such kind of metabolic control and the metabolism of astronaut in human space flight architecture, that would be absolutely enormous progress, because I believe that only in such kind of way, having such kind of strategy and technology, we can explore space beyond of orbit and Mars. Because in the case if something occurs very significant and dramatic for health of astronauts, we can put them in some kind of stasis and for a long period of time with hope that when we return to Earth, we can just taking care about that. Of course, it extends space flight duration, different kind of a logistic. Of course, if in this state astronauts not require food, oxygen, well, it's kind of low payloads, and this is cost for payloads one of significant part of for spaceship. Of course, it eliminates physiological problems. If some kind of conflicts or problem in astronauts, you know, better if you sleep, right? When people sleep, they not argue and have no problems. Yeah. So of course, physiological stress eliminates it. Some kind of oxidative stress, we call it emotional stress. Whatever stress comes from space flight environment, and believe it's a lot of such kind of stress, it could be eliminated if we can reduce metabolism. Of course, protection against radiation, we demonstrate in our laboratory experiment that if animals, most important, not hibernated animals, like we can put in such kind of reversible stasis, they 60% protected in this state because of low metabolism from radiation damage. And this is very good thing. Yeah. Question, of course, remains. Remember, I mentioned that 250 million years bacteria was in some stasis. I was initially very surprised. Okay, let's suppose it's bacteria in such kind of stasis, and we have radiation still on the Earth, right? We can calculate how much radiation damage, most important, not like one-strand DNA damage, but double-strand DNA damage, which is more dangerous and difficult to repair. How many such kind of DNA damage this bacteria can accumulate over 250 million years. And it's come out that even just direct heat You know, not like secondary radiation from free radical, direct hit to DNA, which broke that. It will be so many such breaks that bacteria are not supposed to survive for 250 years, because that already will be dead in that space. But nevertheless, remarkably, and we don't know why, it's absolutely perfectly recovered. So this team of enthusiasts in my laboratory, initially it was 2005, we just proposed this project to center director, and we have his support, and we start an experiment. Of course, with the ambition plan that we will put not hibernated animals, small animals like rodents, in the hypermetabolic stasis. They never hibernate. And if animals not ever hibernate, and usually we're using hypothermia to reduce metabolism. For non-hibernated animals, hypothermia is pathological events. As soon as body temperature, core body temperature decrease, in our case, for humans, for example, a little bit, only 4 degrees, it's severe hypothermia. And we have a lot of problem with heart fibrillation, and it could be absolutely dangerous for us. Nevertheless, we can find some kind of switch which we can turn on, and decreasing of temperature would be considered as absolutely normal and not a pathological event. That was our goal in our research. Of course, we just planned that in 2008 we put rodent in reversible suspended animation, and we did it. We did it, and initially we did it for 6 hours, 8 hours, 14 hours, 24, 32 hours. And for rodent and lifespan, rodent like 2.5 years, it's kind of very significant. Most important, it was so robust program that finally we start to program it. We can just only program automatically mice put in chamber. Its program will reduce, expose it in some kind of atmosphere, which have some agent which induce in hypermetabolic state, and the mice start to sleep. And it sleeps so kind of deeply that, for example, 750 approximately, depending on state, beats per minute of heart reduced to 4. You know, this is significant, 95, 98, sometimes a reduction in metabolism. They require no oxygen, nothing actually. It's beautiful, and it looks like dead, because even sometimes eyes become white, which we related initially to agglutination of gamma crystalline, which protein in an eye, and usually it's considered irreversible. As soon as aggregates form, they never solve. But no, just as soon as you expose mice in normal air atmosphere, probably like 10-15 minutes, and it becomes perfect. And I will show you this slide. Of course, ambition plan presented in this slide was, well, someday we can put humans in such kind of stasis. And we still believe that it's possible. But at the beginning, we create very simple technological, we'll make very interesting design, which I believe now still possible to incorporate in even such project like cubic satellite. Because if you want to prove that proof of concept, let's suppose, yes, if we reduce metabolism, we don't have loss of bone, we don't have muscle atrophy, and we protect the radiation. How to prove it? We will take just hibernated animals, one in active state, group of animals, let's say, another in hibernated state, and send it in space. Let's say for 3 months or even months. If we count that approximately like 2% of bone lost in 1 month, for 3 months they will lost 6%, which is pretty measurable, right? We can determine that. So we return it back and compare one group to another. If we can see that really in hibernated group there is no muscle or bone loss, that will kind of prove concept. And it's very simple. We can put in, let's say, dormice, which I mentioned hibernate 370 days. You know, that's very nice, right? We put them, and they're so small that you can put in your cubic satellite, 5 of them for hibernated state, 5 control active state. Yeah, of course, for 1 year you need to provide a supply for active state, but for those which is not, you don't need to provide nothing for 1 year, even more. And then it's durable. We can immediately see whether it works or not. If it's proof of concept, really could be justified, we can just proceed further. So we create such kind of and design instrumentation which allow us to monitor metabolic state of animals. First of all, we implant some device which allow us to monitor cardiac activity, even brain activity, core body temperature, whatever necessary for metabolism. It's special training. Actually, my guys was working with surgery successfully. When we expose this to hypermetabolic mixture of gas, slides show that core body temperature dramatically dropped from 37 degrees to 10% in this 10 degrees Celsius in this case, but in reality we put even deeper to 4 or 3 degrees Celsius. It's very deep stasis. In this case, no oxygen consumption, as you can see from this slide, no CO2 production in this state, and it's reversible. Heart monitoring, yeah, like I mentioned already, 750 beats per minute you can see, and suddenly you have only 4. Of course, I specifically present this slide which demonstrate heart failure during recovery from this state, and this is very important. No problem to put any animal in this state. Problem is how to recover it, how to reverse it back. And as soon as what we've noticed, as soon as temperature start to decrease from 4 degrees Celsius, for example, in stasis, to recover somewhere around 15 degrees Celsius, we have very significant alteration in physiology. And in many cases, at the beginning, it was heart, which is more sensitive to actually alteration, such conditions. But eventually we develop this very good we can handle, and finally we have 98% recovery from stasis. And this is amazing because in natural hibernators, it's in some cases 60% not survive. We monitor immunological response, for example, because we know that for non-hibernated animals, initially hypothermia considered, as like mentioned, as a pathology, and response is normal. Many inflammatory mechanisms start to develop to protect against this. What we found in these slides demonstrate when we use lymphocytes that organisms could be protected against radiation, and we monitor this specifically using double-strand break in DNA, which demonstrate that in stasis, in deep stasis, these breaks actually probably occur only few of them compared to control. And this is gene expression. It's very kind of interesting how gene response to adaptation to different conditions, to stasis and return back. We're working on this because it's amazing. Why just kind of one of the reasons why we select non-hibernated mice, because in general picture we believe that we can slow metabolism, but at the same time we can increase metabolism. You know, metabolic control means that you're working in both directions, yeah? And the recent study demonstrate that yes, we can actually even increase metabolism. Well, simply, well, you open any journal, Women Journal, and you can see Women Journal that, oh, it boosts metabolism, yeah, it will help you to reduce weight, whatever. But question is that maybe strategy not so well. In the long term, maybe better to reduce metabolism a little bit. Oh, first of all, you can live longer, because we know animals which have such kind of capability to reduce metabolism live much longer. Mice normally lifespan 2.5 years, you know, but mice or bat which living in cave and capable to hibernate, living 49 years. You know, 2.5 for same creature and 49 years, it's something. It certainly demonstrates that yes, capability actually to reduce metabolism can. Well, for example, dorm mice, which I showed such kind of size, it's living 9-11 years compared to normal mice. It's amazing. So this slide demonstrates recently that scientists can even make one gene substitution or modification, let's say, in mice and convert it to super mouse. And this is amazing. What means super mouse? Well, it's probably a lot of efforts, yeah, what should be changed, but it's only one gene modification. It's nothing. You can do it with genetic technology, which we have now, very simple. What they demonstrate, they make such kind of changes in alpha amylase, I guess, muscle protein, grow next generation mice with such genetic modification, and demonstrate that yes, suddenly metabolism change so much that it's so impressive. And on YouTube you can see video. If I have such kind of possibility to turn on this video, you can see this super mouse, which amazing. It's just running like 6 miles without any kind of rest, have reproduction in 3 years. I mean, perfectly, when normal mice like 1 year it becomes kind of questionable. It's of course more aggressive, more strong, living longer, and this is only one gene modification. So it's telling us that yes, probably we can have some kind of room to influence metabolism in both directions. If we want, we can slow down and put in something what allow us to protect, and at the same time we can make super mouse or maybe potentially Superman in this case. So application, potential applications already now we can see everywhere, even in clinical science, in each emergency room. Not each emergency room, but I believe in UK it's already kind of rule that paramedic, this vehicle should have some device which allow to slow down metabolism of brain and prevent against such kind of events as a heart attack and stroke. And the question is that yes, if we decrease temperature, and this slide demonstrates a very simple device which allow you to put 2 tubing in the nose, and we know that distance from nose to brain is approximately in humans like 7 centimeters only, yeah. And if cold air start to circulate, brain start to cool, and hypothalamus and hippocampus becomes cool, and it's kind of help enormously against, for example, stroke, because in events of hemorrhoid, stroke or whatever, we don't have sufficient oxygen for tissue and cells start to die. If you slow down metabolism of cells, they not require any much oxygen. They can survive even longer. In Johns Hopkins University, brain surgeons, when they performing operation on the brain, I was surprised that they can cool down this brain to 13 degrees Celsius and give them not 4 minutes time frame when they should do some very sophisticated operation. It gives them hours to kind of do many operations and return successfully back. So they're using hypothermia actually widely now, and I put here just a couple pictures which demonstrate that yes, even just in New York emergency room, they already using this. Potentially for future, of course, yeah. We believe that eventually we can reach such point when we can reduce metabolism of humans to such kind of degree that we can probably put them in stasis for many years, well, maybe 100 years, but you can ask why. Oh, it's, of course, it's an emergency situation. If nothing could be done in oncological or some disease, a little disease, what you can do? Probably better if you gain some time and to address this problem in future. And we know only kind of area or field now is cryonics, right? But people very suspicious, of course, yeah. There is no any justification or any evidence that after freezing to liquid nitrogen, any creature, except of course, which I mentioned, and its special state. When I mentioned, remember that water bear, for example, can survive liquid nitrogen. Problem is, I did not mention one thing. They can do this only in state of fortification when they dehydrate the organism to remove all water. And in this case, of course, you can decrease temperature to minus 196 degrees, what we have in liquid nitrogen, because there is no water in cell, because water in cell will help crystals, which destroy cell membrane and cell environment. It's also crystallization will create disbalance, ionic disbalance in cell, like calcium, which very important mediator. But recently, like 3 months ago, I guess, so maybe 4, we have some kind of research in Japan which giving us some kind of promise. And it's very surprised that they found some leech which parasite on freshwater turtle, I guess, which can survive liquid nitrogen condition. And leech is significant creature, and it's not dry. You know, it's absolutely full with water and liquid. And they can put in liquid nitrogen and can survive 36 months. Most important, they can make this process reversal. Many times they can warm and cool back. So it's amazing. It's first results which giving us probably it's still potentially possible to have some kind of mechanism which could be developing countermeasure to prevent against frozen tissue. So I believe that sometime we can just put in liquid nitrogen. Most important, which I forgot to mention, that in this stage there is no any cryoprotectors in blood like glycerol or sugar, which normally used as a cryoprotector in another organism. And soon, for example, Arctic fish, to work at 4 degrees Celsius or frozen almost water, they have in bloodstream some kind of sugar or glycerol antifreeze protectors. In this case, no. And this is amazing. What is that? Should be. If we can find, I would be happy to investigate such kind of a creature to find out what is that I can apply for mice and decrease temperature not to 4 degrees to liquid nitrogen. Again, question very interesting. If we decrease metabolism, what happens with brain? Because we know that during long operations, surgical operation, for example, especially old people, for example, operation 8 hours, and then under general anesthesia. Under this anesthesia, usually core body temperature decrease significantly, like several degrees Celsius during operation. So they in hypometabolic some kind of stasis. When they wake up, some of them have some loss of memory for some significant time. And this is only for like 4-7 hours. You can imagine if they would be there months or whatever, how we can show that they still have the memory. And this is a very great question, whether in hypometabolic state we can preserve intelligence, consciousness, and memory. That's very important. At the same time, it's surprising when they investigate Arctic squirrel. And this is a creature which during winter time can reduce core body temperature and be in hypometabolic status a long time. And core body temperature, I guess, record is minus 2.9 degrees Celsius. It's actually frozen, like frozen fish from freezer, you know. It's surprisingly, during this process, squirrel in brain lost 40% of neuron synapses, maybe up to 60%. It probably doesn't tell you much, but in reality I can tell that if you lost 10%, you probably experience significant memory loss. For 60%, people still wondering how they return back, how they reconstruct their memory.